Mais bon, aujourd'hui, on fait mon collègue. So today we have my colleague Shangpeng Sun from McGill University. Shangpeng is one of the grad people with two PhDs. So one PhD from the Beijing Jiangnan University in engineering, and second PhD at the University of Georgia in biological sciences and agriculture. Then he has done one, forced postdoc because he had this, the position McGill, but the COVID hit and couldn't travel, so he has to do a postdoc at the University of Georgia, and then after the travelers region could come McGill and started as an assistant professor in the bioresource engineering department. Thank you. It's yours.
Thank you. Okay. Can you hear me now? Good. Okay. Thank you. Thank you, Jawad, for your invitation for today's seminar. As the introduction by Jawad, so now I'm assistant professor in the Department of Bioresource Engineering at McGill University. So my work is mainly focused on the develop the technologies about plant phenotyping. We develop this, for example, computer vision, remote sensing, is platforms, hardware, and software systems. So today, I'm going to talk about something about multi-scale 3D plant phenotyping from the field level to the organ level. So my lab here today's topic is about 3D phenotyping, but my lab also does something about 2D image processing. I have several slides at the end of this presentation, so I will introduce something about 2D. But today we will mainly focus on 3D. So here, this is the outline. So I will introduce several projects we have done during the past several years. Some of them, that's my part of them, that's from my PhD project, and part of them, that's my work at McGill. So here we have 4 different projects. So for this one, the first is about the field scale. So that's about the field scale plant canopy growth monitoring. And the second is about the root mapping. And the third is about the plant node detection. And the last one, that's about the plant architecture analysis. So these 4 different projects, they are for 3D. And as I mentioned, I have several slides that's about 2D processing, 2D data analysis work. This is the main topics. And first, some background information about our work. So maybe you all know this background, right? So now we have to improve the agricultural system from over the worldwide, because we have the rapid growth of their water population, and also we have the problems about the climate change. So we have to improve our agricultural system. So to improve the crop production or yield, so the phenotyping is now a bottleneck, major bottleneck for this part. So traditional way we see this is soybean here. If we want to measure some phenotypic traits, so the traditional way is send some people or students, or professors, and to the field, and then we use this ruler to measure, for example, height and leaf area, these things. So we know this is not interesting and very time consuming, and sometimes it's the measurement was not very accurate. So recently, over the past several years, starting from 2010, I think, and then more and more research groups, we tried to develop this remote sensing or imaging-based method for the plant phenotyping. So compared to the traditional ones, we call this high throughput plant phenotyping. Here, this figure, this picture figure, this is one of our work, and then we developed this 2D image processing using deep learning method to count this is cotton to count the cotton seedlings. We use cell phones or different platforms to take videos or images in the field. And then we use image processing method to count how many seeds. And then we use this information, we can evaluate the seed quality. But this is good, but you see here, this is a 2D image processing method. So for 2D, we do not have the depth information. And so for this part, it's hard for us to address the occlusion problem. So something, if it's behind the leaf or behind the architecture, we cannot see it. So that's why we need to develop the 3D-based method for in-field or indoor high-throughput phenotyping. That's the background about my work. So first, the first is about the plant canopy growth monitoring. So we know that it's very important for us to periodically monitor the canopy level, the development and growth. For this, we need to periodically to monitor this. And then our first project is about how to develop a field-based platform to do this is cotton plant monitoring. So for this project, we used agriculture-based, this tractor-based platform. This is a tractor, this is this is the tractor we used before. This work is from Georgia. So we work on cotton because cotton is a major crop in Georgia. So for this tractor platform, we developed this enclosure, and inside we installed many different types of here. First, we have the depth RGBD camera. We have the color camera. Also, we have thermal, type spectral LiDAR. We have these sensors. For this project, I used the LiDAR. So this is a 2D light scan LiDAR. Here you see I mount the LiDAR here, and then we have RTK GPS here. So we developed the LabVIEW-based data collection system. So the system can collect the LiDAR scans and the GPS records. So based on this, we tried to reconstruct the 3D model of the plant canopy. So this video shows how did this system work. This is our experimental field. Oh, sorry. Mm-hmm. This is our experimental field in Athens, Georgia. So we mount the LiDAR inside this canopy. As I mentioned, this is the GPS, RTK GPS. So every week we went to the field and then using this system to monitor their plant growth. So you see here, when the tractor was moving in the field, and then the LiDAR, we collect the data for the LiDAR and the GPS. And we use this to reconstruct the 3D models of the canopy. So for this one, maybe you see, because at that time, the LiDAR we used, the model is SICK 911, I think. The resolution is about 1 or 2 centimeters. So it's not high enough to see the more detailed information, for example, the leaves, the branches. For this one, you see, we can just see the overall, the canopy. So once we have this point cloud, the 3D point cloud, and then next, what we needed to do is to develop this point cloud processing algorithms to extract the meaningful information from the data. So first, based on the layout of the field, we can do this, the segmentation. We can detect these plots. So each plot that's around 3 meters long, and we have around 30 plants in a plot. Once we have this individual plot, and then we can first to remove the ground. This is the ground, and then this is the point cloud for the plant canopy. Once we have this canopy, so we extracted 3 phenotypic traits. The first one is about the height. So based on this point cloud, we can generate the height profile, and then we can detect the height, the maximum height and the lowest height or the average height, all these things. And second is about the area. The area, that's the projected area. So because this figure, you see that's from the top-down, top view, and then we can detect the boundaries for this canopy. Once we have these boundaries, and then we can calculate the projected area. And this one, this is the volume. This volume is used the 3D processing method, the convex Hull method to calculate the volume. So for each plot, we can calculate these 3 phenotypic traits, and every week we skip the plants. So we will have the time series data, the 3D point cloud data. After that, so we built these mathematical models. We want to predict the plant growth in terms of these 3 parameters. So here, the first one, this is about the height, and here the second is about the projected area, and the third is about the volume. So this mathematical model, what we want to do is that, so if we have this mathematical model in the future, we can predict the plant growth, and then we don't need to wait until the last minute to evaluate some phenotypic traits for the breeding programs. Okay, and the second project is about the cotton ball mapping. So for cotton ball, you see, for cotton ball, this is a very important because it's directly related to the yield. So mainly these plant breeding algorithms, these programs, they tried to breed some new varieties to produce more yield. That's the cotton ball. So for this project, we developed this 3D best method to count the cotton balls. So usually our collaborators told us usually if they want to get this information, so in the summer, they hire some undergraduate students and then let them go into the field to manually count how many cotton balls. And another thing they needed to do, call it cotton ball mapping. So they needed to count each branch, how many cotton balls. This is a very important phenotypic chart for the breeding programs. So we talked to their students, they definitely don't want to do this kind of work. So that's why here we tried to develop this technology. So this figure shows, this is a 3D model, not 2D. So we developed a multi-view imaging system to generate these 3D models for the plant. So here we tried 2 different methods. The first is about the multi-view imaging method. So we used the same tractor-based platform, and we mount multiple cameras inside this enclosure. So here, and the tractor can go through each row. For each plot, this plot, 3 meter length, this plot, we can generate around 150 to 200 images. So once we have these multi-view images, and then we can use the struct from motion, these algorithms. Actually, we have software to finish this work. So, and then we can get the 3D models for the plants. So, and the second method we used is about the 3D laser scanner. Because for the image-based method, especially under field conditions, the sunlight is a big issue. If the sunlight in the noon, the sunlight is very strong, sometimes you will find that the image quality is low. That's why for this tractor platform, we have to add this enclosure. So the second method, we tried this laser scanner. This is a 3D laser scanner. This laser scanner is an active sensor. Active sensor, that means inside this sensor, we have a light source. So this camera can send some this pulse, and this pulse can, if hit something, and the pulse will be reflected. The sensor can detect this reflected sensor and then measure the distance. So using this principle is called TOF, time of flight. We can measure the distance between your sensor and your object. So, and then we can do the 3D reconstruction. For this sensor, so it can rotate in 2 directions. One is the horizontal, the second is the vertical. For the horizontal, that's 360 degrees. So you put your sensor there and you can scan the 360. And for the vertical, that's 300 degrees. So if we mount the sensor high enough, this sensor, this vertical can cover all these plants. So this is our experiment field. So this is, we have around maybe 400 or 600 plots in this field. And firstly, we need to design some algorithm to select the best position to put the sensor. For example, here, and then we select the positions, and then we put the sensors in this field. And then once finish all this individual scanning, and then we can combine them all together. We call it point cloud registration and generate the point cloud for the whole field. And then once we have the whole field, and next is we developed this point cloud processing algorithms to count the cotton balls or detect the cotton balls. So yeah, we have two methods. We tested the algorithms, this data collection method for 2 years. Overall, it's very robust, it's stable. Here, for the post-processing, so for this, for example, this plot, so if we want to detect individual cotton balls, the first thing is we need to segment the cotton balls from the ground and from the branches. So this is the first step. So in order to do that, so here for the ground, it's relatively easy because we can use the height. We can use the height filter to remove the ground. But for this one, the cotton balls and the branches, because of the sunlight issue, you see some of these branches, you see the color is similar or the same as this cotton balls is white because of the sunlight issue. So generally, we can use the color feature to do the segmentation, but because of this problem, we cannot just rely on the color issue. We need to think about some other features, and then based on these features, we can do the segmentation. So here, based on the observation, cotton ball color is white, and this is brown, good. The second is, we think about this is we call it shape features. Because you see here, for cotton balls is a kind of sphere, but for the branches, it's a kind of stem. So in terms of their shape, they are different, right? So next, what I want to do is to develop some shape-based feature vector for the segmentation. So how to do that? Point clouds. So for the two images, you see each point we call it a pixel. That's just one point there. For point clouds, this unit is a point. So for point, we have the color information, but we do not have the shape information, right? So if we just do the shape feature extraction based on the point cloud itself, that's not a good way. So in order to solve this problem, we firstly generated, we call it super voxels. Super voxels, that means if several points, we have the similar properties, for example, the color, the distance, these features, they are similar, and then we combine them together to generate a super voxel. We call it a super voxel. So here I have two examples. One, this is a super voxel. They combined many of these points together and generate a super voxel. This is for a branch. And this one, this is for a cotton ball. So usually for each cotton ball, maybe we have 10 or even more these super voxels. So now next we process these super voxels instead of their points. So for these super voxels, we have the color and we have the shape, right? So next is how to develop the shape feature, shape features. So for here, because each super voxel actually it's many points that together. So each point we have X, Y, Z, and RGB. We have this 6-dimensional information. So here for shape, I just used X, Y, Z. So if we have many points, and then each point we have X, Y, Z, actually in mathematical way, it's a kind of a matrix. That's M times 3 mathematics. M is the number of the points, and 3 is the X, Y, Z coordinates. So for this matrix, we can calculate the eigenvalues. So For the eigenvalues, and we found that it has a property like this. So the matrix, as I mentioned, is m times 3. So we have 3 eigenvalues. That's lambda 0, lambda 1, and lambda 2. And we found that if the lambda 0 is much bigger than lambda 1 or lambda 2, you see here, the feature is a kind of this stem or branch. And if these 3 eigenvalues, they are similar in the same level, and then this feature can be used to represent the sphere. So this is very good to represent the features about the branch and branches and the cotton balls. So this is the first feature vector. And the second is about the histogram distribution, about the distance between the center point and all these other points. Because for a sphere, for a cotton ball, a sphere, you see, if you find the center, ideal situation, all these the points, the distance between the point and the center point should be the same, right? Ideal situation. But for this one, for a branch, if your center is here, the distance between the point and the center, they varied a lot. So this is also a good way to represent the shape feature. So in my research, I used these 2 features. One is about the eigenvalues, then a second is about the distance. So here, because of the time limitation, I will not give you much about the mathematical description about this one. So once we have these 2, we have the shape feature and we have the color feature, and then we can build a feature vector combining these 2. And next, if we want to do the segmentation, for this project, I used a traditional machine learning method. The traditional machine learning method, one of the features is that you need to build your own feature vector. So I already did this. So I trained a SVM that's called support vector machine. That's a traditional machine learning method widely used in different applications. So we trained this segmentation model, and then we applied this model to segment the cotton plants, the cotton balls, and the cotton branches. So here, this figure shows the demonstrated the results. So for this one, you see, here, this branch, although the color is white, is the same as the cotton ball, but because we have the shape features, and then this method can successfully segment the cotton balls and branches. And next, this is the segmentation result. We removed the ground, we removed the branches, and now here, all these points are cotton ball points. So you see, we have many cotton ball points. Next task is how to detect individual cotton balls. So this is a cotton ball, this is a cotton ball, but here you see many of them physically, they are connected. So if we want to detect these individual cotton balls, one of the challenging problems is to, how to count the cotton balls for this, we call it clusters. Clusters means several cotton balls, they are connected together to generate a bigger cluster. So next task or goal is to detect individual cotton balls from this point cloud data. Here, first, I divided this point clouds into 3 different categories. The first is about, we call it noisy clusters. If we found that this cluster or this one, the volume is very small. We just think about this is a noisy cluster. We can just ignore it. So we don't count this. And the second is about the single cotton balls. For example, here, this is a single cotton balls. And second is about the single cotton balls. And the last one is the multiple ball cluster. So we used the 3D DB scan algorithms to generate these clusters. For this one, it's easy. We can just count this is an individual cotton ball. And the next challenge work is how to divide it or extract it, how many cotton balls inside this big cluster. So in order to solve this problem, my method is about that. First, we calculate the volume for this individual cotton balls or single cotton balls, and their volume about the multiple cotton balls. So once we have this one, we calculate the average volume, and then we can use this volume divided by this, VM divided by VS. So we can estimate how many individual cotton balls inside this cluster. So first we get the number, but we need to know the boundaries for each individual cotton balls, right? So in order to, yeah, K is the number we estimated inside this cotton, inside this cluster. Next is to detect the boundaries for individual cotton balls. For the boundaries, we use the algorithm called K-means. K-means K, you have the K. This is what we estimate how many numbers inside these cotton balls. And then the K-means algorithm is another machine learning algorithm can help you extract the boundaries. So here I didn't show that step. And this one, maybe I can play it again. Sorry. Oh, I cannot play it. Okay. For this one, we introduced the method. This is the results. We used our method to detect individual cotton balls. So first you see the algorithms, we can first detect the individual cotton balls. And the second round, we can detect the cotton balls inside the big clusters. So here, the detected one cotton ball number is 312. We manually went to the field to count how many cotton balls. We selected we did a 2 years experiment. Each year we selected 30 plots. For this one, the ground truth is 311. And then our algorithm detect number is 312. So this is very close. Actually, I did similar work, but using 2D image-based method. So compared to this 2D, the 3D method, the detection accuracy is much higher. I have a figure here. So you see, we did the correlation analysis. So this is the algorithm measured cotton ball number. This is the ground truth number. We manually count how many numbers for 30 plots. The R-square value is around 0.92. So it's much better than the method we used using 2D imaging-based method. So once we have these cotton ball numbers, as I mentioned, we can, to do the yield prediction. We can build a model to predict the yield. And the second application is about this. This is more important actually, because using this method, we know the exact position for each cotton ball in the 3D space. So based on this information, we can do the 3D spatial analysis about the cotton balls. Because some of these breeding programs, they want their cotton ball, that grow like this, very narrow. So because these plants will use less space. So for this one, we can use our method to analyze the 3D spatial distribution analysis. But for the manual method, it's harder to do that. So this is one of the advantages. Actually, later we found that we collaborated with another professor. He was working on the robotic arm for the. So for this one, for our algorithms, and then because we detect individual cotton balls, we can provide the XYZ information to the robotic arm. So we can guide the robotic arm to do the picking. Yeah, this is another potential application for this method. So first, we can do yield prediction, and second, we can do 3D space distribution, and we can help develop the robotic picking. So this is about the cotton ball. For this one, this is scaled. This is the plot level, and the third is about the plant node detection. This work I did part of this work at UGA, and then I didn't finish all, and some part of this work I did at McGill. So for this one, this is the plant node. So yeah, I assume you already know this. This is a very important traits for the branch breeders. So when I was talking to our collaborator, that's a professor who is working on the cotton breeders, and he told me that he was struggling for this many years, and he said that if you can help us solve this problem, I will love you forever. Yeah, so that's why I spent some time for this project. So for this one, because for cotton, actually, you see here, each branch, we will produce 3 or 4 cotton balls. And then, so if the internode distance is short, the cotton plants will have more branches. That means we will have more yield. So that's why that's why here the node detection is very important for the cotton breeding programs. So for this one, yeah, this is the data we collected in the experimental field conditions. We used the 3D laser scanner, and we, as I introduced for the cotton field, we select several points, and then we put the sensor there, and then we have the data, the 3D point cloud data for the whole field. So once we have the data, first we do based on the layout information, and we did the segmentation. We take individual these plants, for example here, but we found that the laser scanner, the resolution is very high. At 10 meters away, the resolution could be up to 1 millimeter. So for each cotton plant, we found that we generate around 5.6 million points. That's a lot. So we cannot directly process this data. So what I do is first use some 3D processing algorithms to calculate the 3D skeleton for the cotton plants. So this is the 3D skeleton. This is the original point cloud. But you see here, the point number is 4,220. That's much lower than the original point cloud. So if we process this one, it will be much easier for us. But you see here, although the data points is much less, but the structural or topology is the same as the topological features is the same as the original point cloud. So this is the second step. And the third is that, so you can see this is the point cloud room leading area. We keep the structural or topological features. And the next is that if we want to detect the nodes, first step is we need to detect the main stem. And then once we have the main stem, we need to detect the individual branches. So once we have the branches, the joint point between the main stem and each branch is the node we want to detect. So finally, we will have the detection results. So here, I will give more information about these 2 steps. The first is about the main stem detection. The second is about the branch detection. For this main stem detection, we used the computer science algorithms called the graph. So this is the original, the 3D skeletons, that's individual points. And then we, based on these points, we generated a graph. Graph is a computer science terminology or concept. So this is the graph we counted, and then we detected the root point. Once we have this root point, we can count how many paths from this point to all these terminal points. And then based these terminal points, we assume the highest point for this path is with the main stem. So we can select the path here, for example, 1, 2, 3, 4, 5, 6. We detect this as the main stem. So using this method, it can, for most of the situations, we can successfully detect the main stem. But we found that because in Georgia, almost every year we have hurricane. Some these cotton plants will be lodged or fall down. For that situation, this method cannot, deal with that situation. That's a kind of limitation for this method. So this video shows, how to detect the main stem. First, we detect the root point, the lowest point. In the algorithm, we call it root point, but it's not a real root point. Once we detect this point, and then using the method we introduced, we can detect the main stem. So once we have this main stem, next is how to detect individual branches. for these branches, with cotton, the branches, they are very long. So sometimes if they are grows together in the field condition, the cottons, they are mixed with each other. So it's harder to detect the whole branches, because here you see the branches is very, for example, it's very long. If we have another cotton branch here, a cotton plant here, these two branches will be mixed. It's hard to detect the whole branch. So what I do is to first, we do a pruning. So this is, for example, this is our cotton branch. This is skeleton. And then we generate a circle with the diameter or radius is R. And then we can remove all these points outside of this circle or this calendar. And then only keep the branches here. So for the diameter here, I used 5 or 10 centimeter. And then once we have these branches, now it's much easier for us to count how many branches here. So in order to, for this one, once we have these branches, I used the 3D DB scan to similar to their cotton ball detection to do their clustering. So we can detect these individual branches. Okay, so for this one, we detect the mass stem, we detect the branch here, so we can find the joint point here, and this is the node we want to detect. So using this method, we can detect the nodes. So here, this is the one of the example I did. So this is the whole cotton plant, but that's a 3D skeleton, and then we detect the nodes. So once we have these nodes, actually based on this 3D skeleton, we can do a lot of more work. For example, this is the node. Once we have node, we can detect the branch angle. And also, as I mentioned, another very important thing the plant breeders they are interested in is to count how many cotton balls for each branch. So based on this work, we can still, to count how many cotton balls for each branch. Okay, then I did some work about the plant architecture analysis. So for the first 3 projects, you see, first we do the canopy analysis, the second is about the cotton ball, and the third is about the cotton nodes. But if we want to characterize the whole architecture of the plant, for example, not just the node, not just the cotton ball, we want to segment all the mass stamp, individual branches, and individual cotton balls. We call it instance segmentation. So we want to know everything about this single cotton plant. So this is what we are doing, trying to do. So here for this figure, you see this is an example. If we have the result like this, definitely we can extract more phenotypic traits. So for this task, I tried 2 different methods. The one is about the traditional computer vision method. So based on the geometrical features or the structural features, I developed some of these algorithms to do the segmentation. For this one, for these algorithms, so here, this is just a rough idea. What I did is first we generate a sphere here with a this radius inside this sphere, and we have, this is part of the plant, right, points, and then we do the 3D circle fitting. So we have the circles here. Once we have the fit circles, we detect the central points for each circle, like this. So once we have these central points, if this is a mass dump, right, so these points, these 1, 2, 3, 4, they should be in a line. And for the branches, because for example, the branches here, they are not in the same line for this one. So we use this feature, we can segment the mass dump and the branches. And here you see this branch, we have this is the mass dump, first order branch, second order branch. We can repeat these steps and then we can segment everything. So once we have this information, these lines, and then we can use this distance information to segment the mass dump and individual branches. So we can achieve the segmentation and segment everything, the tasks. So this is the traditional computer vision based method. Using this one, I tested around 30 plants. So this is good. You see, this plant, this is normal, straight, and have good this architecture. But so this one, this is because of the hurricane issue. This part is called lodged. So, but we found that sometimes our method can still process this one, this plant. Although here you see this is the highest point, or this is the max top, but this is the highest point. Using their own method, we still can do this segmentation. But for some more complex situations, this method sometimes didn't work. So next, we need to think about other methods to solve these complex situations. So then what we do is to develop this AI-based, or we call it deep learning-based, deep convolutional neural networks, this method to do their segmentation. So as I mentioned here, for the traditional machine learning method, if we want to train this model, first you need to manually design your feature vectors. That's sometimes for some tasks maybe it's easy, but for some of them it's really challenging. But the good thing for the deep learning-based method is that the network itself can extract multi-level these features and then combine using these features to do the classification or segmentation or detection. So here the network we used, we call it point voxel convolutional neural network, PVCNN. So this is the whole data processing pipeline. This is the original data we collected using the 3D laser scanner, and then we did some pre-processing. We downsampling, denoising, something this, and then the key part is this one. Is this one. So this is the PVCNN. Usually you find that for the 3D deep learning networks, so because for the point cloud data, some of them, they just directly process their points. But usually for this input one, as I mentioned, we have many points. If you directly process these points, usually you need a very powerful computing resources. And the second, and then the computer scientists, they developed, they proposed this one. It's called voxel-based deep learning. So voxel-based deep learning, that means, so for this figure, for example, we calculate the maximum x, y, z can generate a cube. And then we divide it into small these cubes for each one. For each this small cube, we call it voxel. So this is called a voxelization, this step. Using this method, so it can reduce the number of points because we processed the voxels. But the limitation is that we will miss some information. So this method, we combined these two. We have two branches. The first branch, we directly process their points. And the second, we do the voxelization firstly, and then we use the 3D convolutional operation to process their voxels. And then we combine these two features together and do their segmentation. So for this one, for this branch, so if you see, we have many these mature deep learning networks. For example, the PointNet, PointNet++. These two are widely commonly used for 3D cloud processing. But here we just used the multi-level perception, that's MLP, because in order to save the computing resources. So for this method, we used these two branches, and then we can extract the traits for the segmentation. And then we can do the cotton plant segmentation. So once we have this segmentation, you see, we can first we can detect the mass stamp, we can detect the branches, and we can detect the cotton balls. And for this one, we can extract more these features. So this figure shows, we detected the nodes here, and for each node we have this, the branch angle, and also we can detect the diameter for this node. And also, as I mentioned, if we can count how many cotton balls for each branch. So this way, this method seems perfect. We can extract what we want for these single plants. But the problem is that for 2D image processing, these methods or these tools, they are very mature. You can grab many of these tools for networks for training, or this image annotation tools for the labeling. But one of the limitations for 3D processing is that currently we have several of these networks for 3D point cloud processing, but we do not have these good tools for point cloud annotation. So this method, here, I say this is a fully supervised deep learning network. Fully supervised deep learning network, that means if you want to train this network, first you need to label this data and then use this labeled data to train the network. But for 3D point cloud, the labeling is, if you do it, you will find that it kills you. So it's very challenging. So for this point cloud, for example, for this one, if we have a meaning level these points, so if we do the labeling, so it's very time consuming. Actually, currently we are struggling about this. And then we think about how to solve this problem. This is our a project we are doing, we did here at McGill. So here, starting from here, we work on the soybean, because in Canada, cotton is we don't have this cotton plants, but we have this soybean. So that's why I switched to soybean. So here I have two figures to show the difference between the fully supervised and the weakly supervised deep learning networks. So for the fully labeled point cloud, for these soybean plants, we needed to label individual points. So that's very time consuming. But for this one, this is partially weakly labeled. We just needed to label very small part of this point. For example, we tried 50, 100, and 200 points. We use this limited labeled numbers points to train a network. And if this is successful, it will save our much time, right? So for this one, this is the data processing pipeline. First, we do the multiple imaging to get the 3D point cloud, and then we developed this labeling tool to do the point cloud labeling. And then we developed this point cloud network, point cloud processing network. Once we do the instance segmentation, we can calculate, for example, for each leaf, you can count the leaf length, leaf width, leaf area, branch angle, all these things. So for this one, this is the method. So we have two steps, mainly two steps. One first is called self-supervised pre-training. We need to pre-train a network. This network can extract the basic structural information about the plant. And the second, once we have this model, then we use the limited labeled data to fine-tune this network. And then we can achieve these two goals. One is called semantic segmentation. The second is called instance segmentation. So this figure shows how to do the self-supervised pre-training. This is the network we want. So for this plant, this is the input. We randomly rotate it and get two different positions or geometrical these positions. And for each position, and we can calculate a matrix here. And based on these two metrics, we calculate a matrix called cross-correlation matrix. And then use this, we develop a loss function, and we can train a network, this network, and extract the basic information about the plant structure. So once we have this plant structure, and then we use this pre-trained model, the basic structure information, we add another, you see here, this is MLP, multi-level perception. This network, we can do the semantic segmentation. And also, we developed this part. This is a class method. This is the offset modules. So we use this one. We can do the leaf instance segmentation. So here you see, this is the result. Using this one, the first is about the ground truth. We manually labeled the data. And the second is the fully supervised network. We tried the PVCN, we tried the PointNet++ network, And this one, this is our result. We used 200 points. We labeled 200 points. But you see, we can achieve almost a similar performance for the fully supervised method. The performance is similar to this. But we just only needed to label 200 points. So it will save us a lot of time. So now this is the fully supervised method. So you see, for this 3D segmentation, initially we used the traditional machine learning method. We needed to manually design our feature vectors. And the second is the fully supervised. We needed to label all these points. And the third, now we are trying this weakly supervised method. We just only needed to label partial data. But in the future, maybe we can try self-supervised. We don't need to label any points. Yeah, but that's future work. This is the 3D point cloud. And here next, some slides about the ongoing project in my lab about the 2D. So the first one is about the sensor fusing. So we want to fuse the RGBD, thermal, hyperspectral, all this information, because sometimes if we just rely on RGB, XYZ, it's really hard to get high performance this segmentation. But if you add some other information, could be better. So this work is did by Xintong, my PhD student. It's here. So yeah, this is ongoing project. And the second is about this strawberry fruit counting. This is for this is 2D image. This is a video. So this is work did by Shuhai. He will join my lab next January as a PhD student. So this method is vision-based deep learning network to count how many strawberry fruits. This is for vertical farm, for greenhouse, vertical farm. So, and the third is about the UAV image processing. This is also did by Xintong for her master program. We use multiple spectral images to take these images for the spinach field, and then we want to extract some VIs and then predict the yield. Actually, here, this is some work here. I have several other works about 2D, but do not have time for this work. And last is the acknowledgement. Yeah, so my work was supported by this agents. So in the US, so their work, especially about cotton, they're supported by NSF and USDA NIFA. And in Canada, so projects are supported by NSEC and FRQNT. And also we collaborated these farms, these vertical farms for Shuhai's project, Xintong's project. And also this one, this is RQAD. We have a project with Davad and yeah, for soybean root phenotyping. Okay, I think too much time, sorry for that. That's all for today's seminar. So because of the time limitation, I cannot introduce more about the detailed information about the method. But if you are interested, and you want to know more, here we have our lab website. You can, to check more. And if you have more questions, you can send me emails. Okay, thank you very much.